So today I'm going to talk about whether upregulation of the proteasome can extend lifespan. This is a slide I'm sure that everyone is quite well familiar with now, representing 9 of the hallmarks that are associated with aging. And so today I will be focusing on the proteostasis piece of this puzzle. Now, proteostasis is maintained by several mechanisms within the cell. These include autophagy, proteasome degradation, and chaperones that refold proteins that have become misfolded. And in particular, I will be talking about proteasome dysfunction and whether this can be ameliorated by upregulating subunits of the proteasome. And just an overview of what is the proteasome. It's a multi-protein complex which dismantles damaged proteins and also proteins that aren't needed anymore, and transcription factors. These are found in both the nucleus and the cytoplasm of all eukaryotes as well as some prokaryotes, and they are essential to the cell, including for the cell cycle and gene expression, as well as I mentioned, eliminating damaged or misfolded proteins. Now, the proteasome comes in many forms. 2 of these are the 20S and the 26S proteasome. The 20S makes up the core of the 26S proteasome as well. The 20S degrades oxidized proteins primarily, and this process is ATP independent. The 26S proteasome degrades polyubiquitinated proteins. These include often the transcription factors that are marked for degradation, and the process is ATP dependent. Now, there have been many reports in different species and in different tissues of a decline in proteasome function with age. This has also been reported in worms, flies, honeybees. There's some debate as to whether the 20S function decreases. Some reports have found that the amount of proteasome stays the same, but function decreases. But overall, there is a loss of proteostasis. Now, in Drosophila in particular, which is the model that we're working with, it's well established that the 26 proteasome's function declines with age. And here is showing proteasome activity in the heads from fruit flies. So both our findings and from other labs are that the 20S proteasome maintains its activity into old age, whereas 26S activity is markedly reduced. Now, this is an overview of the subunits. I want to draw your attention to the beta ring, as those will be the subunits I'll first be talking about. In particular, the beta ring contains the 3 catalytic subunits of the proteasome, beta 1, beta 2, and beta 5. An earlier work done in fibroblasts had found that with passage, that many of the subunits decreased by later passage, and in particular, the 3 catalytic subunits were found to be lower in late passage cells. And these authors suggested that it was these beta subunits that are rate limiting for proteasome formation, and that therefore upregulating these subunits might restore proteasome function with age. And they first did this again in the fibroblasts, and they found that by upregulating just one subunit, the beta 5 subunit, they were able to simultaneously induce upregulation of multiple subunits, including the other catalytic beta 1 and beta 2, and importantly, this increased all 3 of the proteolysis activities of the proteasome. They went on to continue this work in C. elegans, and once again, upregulating the beta 5 subunit. Here we go. Increased multiple subunits. It's hard to see this from here. including the beta-2 subunit and beta-1, and then also increased overall proteasome activity. Significantly, this also extended the lifespan of the worms and greatly improved their survival under conditions of oxidative stress. So now we wanted to see if we could replicate this in flies. And as was seen in C. elegans, upregulating the beta-5 subunit called ProsB5 in flies upregulated expression of multiple subunits and increased proteasome activity, both the 20S and 26S. Also, as was seen in C. elegans, upregulation of the beta-5 subunit increased their survival under conditions of oxidative stress. However, we were surprised to see that this ubiquitous upregulation of the ProsB5 subunit did not result in lifespan. There was no effect at all. We went on to upregulate ProsB5 specifically in the nervous system, and this was using a pan-neuronal promoter. So it's upregulated not just in the brain, but in the ventral nerve cord and the nerve system throughout the fly. And this robustly and repeatedly upregulated, increased their lifespan, both medium and maximum lifespan. And we are in the process of repeating this experiment using the ProsB1 subunit. There's a suggestion from the earlier literature that this one should act similarly to upregulating ProsB5. Now, there's several questions we had from these results. As I showed you, upregulating the beta-5 subunit upregulates both 20S and 26S activity. So we wanted to know, are one of these required for life extension, or are they both? So now I want to draw your attention to subunits that are specific to the 26S proteasome, particularly the RPN subunits, 3 of which I'll be talking about are RPN11, which deubiquinates substrates, and RPN5 and RPN6, which are involved in binding the 19S cap to the 20S core to form the 26S proteasome. It was found in flies that upregulating the RPN11 subunit increased only 26S proteasome activity, and also that it extended lifespan. However, I do want to point out that this lifespan was done at 29 degrees Celsius, which is warmer, much warmer than flies are normally kept at. You can see that their control lifespan is shorter than you normally see in the control lifespan of flies. But at this temperature, RPN11 overexpression did increase lifespan, whereas knocking it down greatly decreased lifespan. Looking at worms again, increasing the RPN6 subunit also increased stress resistance, and it extended lifespan, but just like we saw in Drosophila, it's only under conditions of proteotoxic stress. You can see at 34 degrees Celsius, which is a very severe heat stress for C. elegans, there's a large increase in lifespan. At 25, which is a mild heat stress, this increase is still significant, but much less, and then it completely disappears at 20 degrees Celsius. So we are in the process of testing this in flies to see whether ubiquitous overexpression will only be beneficial under conditions of proteotoxic stress, and whether if, like ProsB5, neuronal overexpression will increase lifespan under normal conditions. So we found that overexpression of RPN5 specifically increased 26S proteasome activity. And we now have the lifespans in progress. Flies are quite long-lived compared to the worms I previously worked with. So at day 50, we are only starting to see a difference. And it looks like it will not extend lifespan at regular temperature, but it is really too early to tell. And no difference at all yet with the pan-neuronal overexpression. So stay tuned for those results. And the other question that we had from what we observed with the ProsB5 overexpression in flies is, why does ubiquitous ProsB5 overexpression not extend lifespan while pan-neuronal does? And we thought the two most likely reasons for this is that increased proteasome activity might be detrimental in some tissues. Also, it may be that increased proteasome activity is truly important in the neurons, and the particular promoter that we've used expresses ProsB5 much more strongly in the neurons than does the ubiquitous promoter. And this is just showing both the 26 and 20S activity in whole fly lysate. And so you can see that this is the ubiquitous ProsB5 overexpression, which is in all cells, but then the pan-neuronal overexpression, which again is just in the nerve cells, causes a much greater increase in both 26S and 20S activity.
This was done with the heads, or?
This is whole body. And again, I just want to remind you that the pan-neuronal is throughout the whole body. When I first did this experiment, I was thinking it was primarily in the heads, and this result surprised me. And having previously worked with C. elegans, I immediately thought of cell non-autonomous, where almost there's so many different things where you do something in the nerves that communicates to the gut, and then they live longer. But that doesn't appear to be the case with the flies. One of the tests that we do with the flies, besides measuring lifespan and proteasome activity, is looking at their movement, their spontaneous activity throughout the day. This is just a little schematic of how we do this. We have the flies' vials in a machine that has infrared lights across at specified time intervals, and so then records how many times those beams are broken. From this, we can get information on circadian rhythm and also spontaneous activity. And I want to point out here that at middle age, or day 50, the flies with ubiquitous ProsB5 overexpression are already showing significantly less spontaneous activity than the control flies. This suggests that overexpressing ProsB5 ubiquitously is indeed detrimental, perhaps in the muscles or cardiac, I'm not sure which tissue yet. Whereas even at old age, with the ProsB5 pan-neuronal overexpression, there is no difference in activity levels. I want to remind you that overexpressing ProsB5 ubiquitously does improve survival under oxidative stress. I wanted to repeat this with the neuronal overexpressed ProsB5, and there was absolutely no effect on stress resistance. And so this, from what we've done so far, I conclude three main points. One, that whole body upregulation of the proteasome does increase stress resistance, but it has no effect on lifespan. This is similar to what has been found in C. elegans. And it appears that the effect of proteasome upregulation on stress resistance and lifespan are separable and tissue-specific. Finally, pan-neuronal upregulation of both 20S and 26S proteasome robustly extend lifespan in Drosophila. We are still investigating the mechanisms as to how this happens. So some of the future directions are in progress. I'm continuing to look at the health span of the flies with pan-neuronal overexpression of ProsB5. I'm looking at their climbing ability and spontaneous activity and circadian rhythm, as I showed an example of on an earlier slide. And then I'm repeating the lifespan and these health span assays with upregulation of ProsB1 and RPN5. And then some other experiments that we're looking to do are using a GFP marker of synaptic vesicles to, again, try to get more at the mechanism whereby these flies are long-lived. We want to see if perhaps ProsB5 overexpression prevents neuron loss with age. And also we're planning to overexpress ProsB5 in subsets of neurons, such as just the ventral cord or just the motor neurons, to see if these recapitulate part of the life extension that we see. Again, again, to get a better idea of where is proteasome overexpression beneficial. And that concludes my talk. I want to thank my lab and thank you for being here and for all your support and your help with these experiments. Thank you.